So yeah, thank you for having me today. I'm sorry I can't be there in person, various things getting in the way of that. But it's a real honour to be able to present to you some very recent work actually. You see my sidebar? That's quite irritating. Hopefully you don't see that. Mind, that might go away. So a bit about me. I'm from a background of environmental sciences. I did my undergraduate in physical geography actually at Bristol. Then I did, I specialised in sort of research areas of life in cold environments during that degree. And that got me really interested in extreme microbiology. And that led me to do a PhD with Charles Cookell. At that stage, he was just moving up to Edinburgh, so I moved up there with him. And that PhD was focused on the potential for life on Mars that can make a living from iron redox metabolisms. And that sort of got me interested in microbe-mineral interactions, and that saw me move here to the University of Manchester in 2015. And I've done a number of different postdoc projects here, and then secured my own funding, firstly as a NERC fellowship, which is what most of what I'll present today has been done through, and then latterly also a University Dame Kathleen Olorenshaw fellowship, which is tenure track, and a BBSRC David Phillips fellowship. And I'll talk a little bit about the research that funds towards the end, because it's only really just kicking off the ground now. So I've just moved from down the road at the Williamson building, which is the main building for Earth and Environmental Sciences in Manchester, up to the Institute of Biotechnology, where the kind of approaches that I use, which I'll talk about here today, have got some exciting potential to biotechnological applications, and that will feature in my talk as well. So with that, I'll get straight into it. So today I wanted to talk about life in the engineered subsurface, and then how you can use that knowledge and those insights about the deep biosphere to find ways to help us achieve a cleaner, decarbonised future. So first of all, just to introduce what I mean by the deep subsurface. There are lots of different definitions, and it's a bit of a hazy area, but it's helpful to think of it as below local hydrological flow systems, so that being below that level where you have your water table and your other surface connected hydrological features, that cuts off the deep subsurface from the surface area really. And so that can change depending on whether you're talking about the continental crust or the oceanic crust, which is obviously very different in that respect. These environments are almost always isolated from sunlight. Well, they are always isolated from sunlight, and they're typically anoxic. So it really restricts the metabolic activity of life in these systems, which is fueled by chemical energy. So you have lithotrophy. And this fuels a slower pace of life. We get an understanding now that microbes in the deep subsurface might have a doubling time of hundreds or thousands of years, which is obviously very difficult for us to study when we do projects that are only, at most a few years long. So it's a very different kind of biosphere to what we see around us. But it's expected that up to a fifth of the Earth's biomass resides in the deep subsurface. This is an absolutely enormous amount of biomass. And in this deep subsurface, you'll find hotspots of microbial communities and activity around, for example, rock interfaces, or where you might find natural fractures. But the kind of access that we'd need to study the deep subsurface in a pristine environment is very difficult, extremely challenging and extremely expensive. So my interest in the engineered subsurface enable us to have a window into life in the deep subsurface, but piggybacking on what we're already doing down there. So 3 good reasons why you'd want to study life in the deep subsurface, other than it's just fascinating. The first is that the subsurface harbours the unseen majority of microbial life on Earth. You can see this is an updated tree of life from 2018. Most of the foliage on this tree has been brought to light using genome-resolved metagenomics. Many of them, the ones with the dash boxes around them in this figure, contain no cultivated representatives. That might have changed now, but certainly when these whole clades were discovered, these phyla, they were previously what we'd call dark matter, microbial dark matter. So this actually, most of this knowledge in this figure comes from one environmental site, and that's the shallow subsurface environment. It's an aquifer in Rifle in Colorado. And this group, Jill Banfield's group, they plugged away at just characterizing the community that lives in this system, and they, in doing so, uncovered this huge diversity. So this is just one impression of what the diversity in the subsurface looks like, and that's not even the deep subsurface, really. Second reason, and very much related to my talk today, is that the microbiology in the subsurface impacts on our engineering of the subsurface, and therefore impacts on us, both for society and for the environment. And then thirdly, and something I'm moving more towards in the next few years, is the biotechnological aspects of this. So this, I hope you can see my cursor here, but this remarkable diversity that we're only just starting to uncover is more than likely to hold a number of different useful tools that we might want to harness, either for the good of the environment or the good of society or for economic gain or all of those things ideally. So you can think of this deep subsurface biosphere as a repository for potential biotools that we can tap into. So this cross-section of the subsurface gives you an idea of how much we rely on it. We rely on it for conventional oil and gas, but especially in the next sort of couple of decades, we'll rely on it more and more for the disposal of our nuclear waste legacy. And also, a major part of carbon capture and storage is CO2 storage. We also have plans when we start to introduce hydrogen into our energy grid, and we'd need to find a way of temporarily storing that along with compressed air. All of these things rely on being able to use the subsurface. And the role of microbiology, having said what I've just said the previous slide, is therefore very critical to understand. And I'm going to focus a lot now on the microbiology of gas systems, and in this case, shale gas. So straight to the US, because that's where the majority of what we know about particularly the microbial life in these environments comes from. So here you see actually quite an out of date now, this is 5 years old, but a really nice infographic that featured in the Washington Post. And this gives you an indication of oil plays, as we call them, in the green, and shale gas plays or natural gas plays in the pink. And along the top here, you get a sense of how productive these have been in that 10-year time period spanning 2007-2017. And this is based on trillions of cubic feet per year. So just to highlight the Marcellus here, this is in the east area here of the US. This is by far the most productive to date, although I believe the Utica is now catching up. The Marcellus will become relevant in a few slides' time. So just to give you an indication of what actually happens during hydraulic fracturing. So in this snapshot here, we have a depth profile down to about 4 kilometers. In fact, most shale gas wells that have been studied, certainly for the microbiology, are a bit shallower than that, still 2 to 3 kilometers. And here you see that there's a vertical drill down to the horizon where you have the tight gas reservoir or gas shale. So this is a tight rock where the natural gas that we try to recover is locked away in poorly connected pores that are very, very small. So this is why hydraulic fracturing is required to stimulate that rock and allow that gas to flow. So in this case, you see a vertical bore down to the depth of the shale, and then a horizontal bore, horizontal drilling, and then these charges get set off at various locations along this well bore, and water-based fluids pump down at incredibly high pressures to induce fractures in the rock that are bigger than that you'd find naturally. These fractures are then held open by a proppant, which is typically sand, and this stops these fractures from closing up again with the overburden pressure. And once this fluid is recovered, then the gas can flow from the peripheral rock through this well bore up to the surface, and these shale gas wells can produce for decades. So this is a very thirsty process. It typically requires, this is typical for a US shale gas well, I should say, tens of millions of litres of fresh water per pool, per well. Although it's very common now to reuse some of the water that gets recovered from the surface in an attempt to reduce that water pressure. And just to show what this actually looks like, you've already seen this on my title page, but this is a really nice infographic, or this diagram put together by the Pacific Northwest National Laboratory at the time of a major publication coming out a few years ago, just to give it a better indication of how this might look in situ. So this, on this diagram on the left, it's lovely blue, pure water. In fact, this fluid is amended with all sorts of different additives that each have a different function. And there are more than 1,000 individual disclosed hydraulic fracturing additives that are commonly used in input fluid. So just to delve into that a little deeper, this is a very busy slide, but there are two key things to take away from it. The first is that different additives have different functions. So, for example, gel-forming agents, these get added so that you can thicken the fluid and allow the delivery of proppant to the fractures. These can be cross-linked to do that and then broken down. So these are a temporal sequence, these first three. And then you have additives that serve a function throughout the whole process. For example, corrosion prevention, prevention of clay collapse and scale formation, and also here on the bottom right, prevention of microbial biofouling. Clearly they don't work very well, or else I wouldn't be talking about this today. The second thing to pull away from this is that I've just extracted the most commonly used version of each of these groups, and they're almost all organic compounds, and they look quite tasty as food for microorganisms. So that's another key thing to remember. So taken together then, what we understand about this process, this engineering intervention, is that it creates new ecosystems at depth. So what do I mean by that? Well, if we start with the pristine rock, which you see in this red circle here, this shale rock is typically characterized by nanodarcy permeabilities, limited meteoric water. Often in the diagenetic history of these rocks, they've been pushed much higher than the upper temperature limit for life. And in that case, we would consider them to be paleo-pasteurized. Some shale gas is in fact biogenic, but many formations are purely thermogenic. And so in essence, this is a very limited microbial habitat. However, when we start drilling and pumping fluids down well, as well as additives and the microorganisms associated with all of that, we open up this system. We create new fractures that serve as new colonization surfaces. We potentially liberate organic carbon and useful inorganics from the shale. And we know all of this. We know that this serves as a microbial habitat because we can collect these fluids at the surface and we can study their geochemical and microbiological signatures. And in this case, we do that through culturing and DNA-based approaches. And I'll talk about that a bit more. So this is that paper I alluded to, where we had that nice diagram from my title slide that was released to coincide with this. This is a particularly nice study by collaborators of mine in the US who had studied the microbial ecology in succession in one Marcellus shale gas well. So if you remember, the Marcellus is the very productive formation in the US. The key things to point out here is that we have 5 different samples spanning different points along the hydraulic fracturing process of one well. So we have input fluids, then fluids recovered after 7 and 13 days, which are most likely to be flowback fluids, which are just recovering what you put in. And then much later on, we have 2 samples that represent later time series, particularly the latest one, which is almost a year. These colours represent different microbial taxa based on metagenomic data, actually. Normally you see these graphs for 16S gene sequencing, but this is the entire genome of all these organisms pulled out. And the thing to highlight here is the taxa in this red box, which you see prominently in these last 3 samples. So these give you an idea of, especially in these last 2, persistence. These have been persistent in that community over 250 days. And if you see the dots plotted on top of these give you an idea of population size, which is meaningful because it shows us that the enrichments we see over time are true enrichments. So the population is growing, therefore the relative abundances can be considered a bit more absolutely. And just to highlight this red one here in the final time point, this is Methanahalophilus, which is a methane, it's a methanogen, but it's salt loving. But methanogens are notoriously difficult to grow because they're so oxygen sensitive. So this is a really nice indication of the anoxic conditions that you get after about a year since fracturing began in a shale gas well. Some of these key players that we find in this red box were detected in the input fluid. You can see some of them here. So the Marinobacter, the halanaerobium and the halomonadaceae. This star is just to represent that this is the same genome. And then when we look at these major shifts in this timeline here, we see it coincides really nicely with increased salinity. So what you see in this graph is along the x-axis, time since hydraulic fracturing in days, and along the y-axis, conductivity. And the conductivity is such in these systems much greater than the average seawater salinity, and in line with that of the Great Salt Lake, where you'd find halophilic organisms. And this is true across a number of different shale gas wells and formations. So it's about between 50 and 100 days, you get this sharp increase in salinity, which seems to drive the community to a different place. And that was just for the Marcellus, which is one of these wells here. But if you look at other formations across the US, then you see a similar situation in that you get many of these common or usual suspects occurring in all of these systems, and it's a fairly low diverse community. The particularly important one here is halanaerobium, because it ends up dominating many of these communities. So just to talk a little bit about a particular well in the Marcellus, which is one that I had the opportunity to work on. So this is to show you whereabouts we're looking. This is the Appalachian Basin. This is West Virginia right here. And this is the well pad that you can see in this photograph here. And these are there are 5 wells drilled from this well pad, I believe. I think there are 4, actually. There are 4. The names are a bit misleading. And it's this 5H one that I was studying. So it's the longest one. The lateral extent is several miles long. I'm going to mix up my units bit here because I've still not managed to figure it out in my head, but it's about 2.5 kilometers deep and a few miles long. So figure that one out. But it's a really nice system because this is like basically a science experiment in the field. So researchers such as myself, through collaborations, can go and access these environments to study them in detail like you wouldn't be able to do on a purely commercial venture. So just to give an idea of what my role was in this. So the fluids that you've already seen, these were collected over a time series since hydraulic fracturing began of one single well. These had actually already been studied for other things, but what I was working on was the metagenomic data. So in these samples, we have all sorts of minerals, and crucially, we have all the biomass. So that gets concentrated. And you see on this filter here the telltale rusty sign of iron oxide. In fact, when these fluids are recovered at the well pad, they're clear, but they're very rich in iron too, which quite quickly, given its instability in the presence of oxygen, it flocks to form a fluffy iron oxide, which actually helps us because it helps concentrate that microbial biomass. Once you've concentrated that, you can then extract the genomic DNA. And this genomic DNA is we're not just taking out single genes, we're taking everything present. So this serves as a blueprint for what that community can do. Once we subject that to sequencing and reassembly, we can start to put those genomes back together. Once we've reconstructed these genomes, we can start to assess for purity, keep all the good ones and omit all the incomplete genomes. And then we can start to interrogate these genomes using these kind of questions. So who's there? What metabolism are they able to operate? How does their abundance change over time? And therefore, what might they be doing differently over time? And what that might their role in the ecosystem be? So in the interest of transparency, I only came in with the data. The group I worked with already had all this data, sat on hard drives, and I went in to analyze it, and in doing so, learn how to use this technique. But not just the metagenomics matters, we need to place it in context of its natural environment. So this is some geochemical data from there expressing salinity as conductivity over time. And we can see that this is pretty typical for Marcellus. It's about on the same level as the Great Salt Lake. And then we looked at, again, looking at the metagenomes, we looked at what that community profile looked like over these different time periods. And we got a very low diversity community compared to the previous well. So just to highlight, this is from 100 days onwards. We don't have any of that early stuff when the salinity is still increasing. But we have the same pattern of increasing cell numbers over time. And we have a couple of the usual suspects, particularly halanaerobium at this 250-day point. In this case, I've used HF instead of hydraulic fracturing, but I realise that's also hydrofluoric acid, so don't get confused. But But we also found something unexpected, which is not particularly high in diversity. You can see on the right here that it's very low diversity in 2 time points only, but it's something that we hadn't seen in any other systems before. We had an inkling early on this was novel. So this was what I ended up working on with this data. We already knew a fair bit about all these other usual suspects, but this was new. So we started to analyse the genome of this organism to understand who it was, what it could do, you know, what the role in the ecosystem was, and to try and understand a bit more about its heritage. So firstly, we looked at its genome that we recovered from this metagenome. In fact, we did all this work on the one recovered at 313 days. We didn't get the whole genome at 152 days, but we got a signature of it, which is expressed in this graph. So to find out who it was, we were able to recover the 16S rRNA gene from its genome. Now, this is what we'd use as a sort of a fingerprinting tool. When it was discovered as a phylogenetic identifier back in the 70s, it revolutionized microbiology. And that's when we realized that prokaryotes were not just bacteria, they were bacteria and archaea. So it's a very powerful method for at least placing it roughly in the tree of life. And in this case, we managed to find an 89% match to this organism here, Corallia margarita acagimensis. So this straight away gave us an indication of what phylum our organism comes from, and even down to Punicae coccaceae, which is the family. Now, 89% might seem quite high, but in fact, it's pretty low for a 16S identity match. You'd expect something up in the high 90s if you found the same species, maybe in 95 if you found the same genus. So 89 indicated early on that we might be looking at a new genus. And then when we did the whole genome identity, so we took the whole genome of this guy that we found in the system and compared it based on its amino acid sequence to its nearest 16S match, we found a 48% match. And this is confirmation that we had a novel genus. And you can see here, this is my tree of the whole Microbial phylum based on cultivated representatives and from some of the metagenome assembled genomes. And it sits up here in the top just to highlight it, but we got to name it. So in this case, we called it Candidatus marcellius, after the Marcellus formation it came from. So then we can start delving into its genome to understand what it might be doing there. Now, I didn't make a big thing of this earlier, but guar gum is going to become a recurrent topic in the rest of my talk. It's the most widely used gelling agent in input fluids. So it's also used in food. It's a high-grade food reagent. So chances are if you get some chocolate chip ice cream or something like that, it might have been thickened with guar gum or xanthan gum. They're very similar in their function. Essentially, this is what thickens the fluid that goes into the well to deliver the sand to the fractures, and then it's broken with a chemical breaker, and then it's all recovered at the surface. But when you look at the molecular structure of this, we basically have a whole load of sugars. So the backbone is made of mannose, and then we have these galactose side chains. And then in the genome of this novel organism, we found all of the genes required to fully break away the side chains and break down the backbone, so that we're turning this polymer into monomers, and these monomers happen to be hexose sugars, which are a very rich source of energy. And we also found the ability for this organism to use the mannose that forms the backbone in its glycolysis. Often you know that organisms use glucose in glycolysis, but there are alternative sugars that can be used, and essentially this kickstarts metabolism. There is an alternative polymer that gets used in input fluids to guar gum. This is a synthetic polymer called polyacrylamide. It's a different form of fracturing called slick water fracturing, and is in fact what was used for the well this organism was recovered from. It's a more expensive additive in these systems, but it enables, instead of thickening the fluid, it enables you to pump at much higher pressures, so it functions slightly differently. And for this genome in Candidatus marcellius, we found a number of amidase genes that could cleave off this amino group. Perhaps there's some use there. But as you'll see, we also found evidence for some other functions as well. So we found, well, just quickly, we found evidence for nitrogen fixation, which potentially could be an ecosystem service in this environment. Osmo protection was something we were particularly interested in, because this seemed like it wasn't a usual suspect that you'd find, nor did it seem to come from a group of organisms that were adapted to high salt. So we wanted to understand how this particular organism could cope. And this is a snapshot of a much bigger heat map, but it shows you the genes that we found present with colour, absent with grey, the genes that encode for either a saltin, coping mechanism, or compatible solute transport and production. Now, this compatible solute approach allows organisms to basically accumulate organic anions in the cytoplasm, which helps balance that osmotic pressure from outside to within. The saltin strategy is a much more hardcore approach that you find many more halophiles using, but we found genes relevant to both, which was interesting. And I don't want to get too technical with this, but we also found nice evidence for adaptive immunity towards viral predation. Now, I didn't go into this in my introduction, but there's a number of studies, including the one I highlighted, found that active viral predation is an important driver after salinity increases of the community composition over time in these fractured shale environments. So in this genome, we found evidence of two prophage. So these are viruses like these here that have integrated their own genomes into the genome of this organism. We also found a number of CRISPR-Cas arrays. So these are inherent immunity or immune systems that organisms are able to encode. And this is comprising of repeats and spaces. So the number of spaces gives you an idea of the history of their viral predation, and the repeats save some of the genomic information about that virus. So we found a very large number of spaces, and this actually should say no repeats, but we didn't find any repeats that matched to a shale-wide viral database. So in other words, it's been predated on a lot in the past, but from a different habitat. So there was more to that story, and it became a bit more of a comparative genomic story with the rest of the Verruca microbia phylum. So if you're particularly interested in that, then please check that paper out, particularly focusing on glycoside hydrolases and carbohydrate degradation. But what I want to talk about now is shale gas communities closer to home. So this is a summary from a chapter that feels like it's been in press for about 2 years now. But this is to show that beyond that earlier snapshot, which just showed a few systems in the US, we now have insight from other shale gas wells in Canada, and also a couple from the Szechuan Basin. And shortly, well, we have our first paper in review at the moment that deals with the microbiology of UK shale gas wells. So that's what I'm going to talk about now. So this was published 4 years ago. It doesn't seem so long ago, but it feels like a lot's changed in the UK shale gas landscape since then. This was an estimation of resource recoverability, or at least resource size, for the Bowland Hodder Shale, which you can see in this diagram to the right. So it's underneath our feet here in Manchester, and it spans different areas from the west to the east coast. And it seemed like there was an absolutely enormous resource there. I mean, this estimate quotes at 1,329 trillion cubic feet. How much of that is recoverable and economically viable is still unclear. But this led to lots of different exploration licenses being granted, and you can see those very faintly with these black boxes here. And the only company to have explored or done exploratory fracturing in these wells is Quadrilla. And I actually gave a talk about shale gas microbiology on the day that Quadrilla posted this on their LinkedIn profile, and it was a picture of them starting to do fracturing at their Preston New Road site. So this is near Blackpool, towards the west coast here. Now we have a moratorium on fracking since November 2019, which I'm sure you're all very aware about. It seems to change every day, and there seems to now be lots of people pushing for that to be reversed, but I don't see that happening. But nonetheless, I was lucky enough to get the opportunity to work with Quadrilla and use their samples from their well to characterise the communities in them. So we now have what I'm about to show you is sort of the first insight into life in the deep subsurface of the UK. Just to give you a very brief timeline here, these wells were drilled in 2018. The drillers are different to the fracturing crews. So the two wells were drilled at the same time, but weren't fractured at the same time. And I managed to get hold of some drilling mud from the first one, which we've called Bowland 2 for reasons of the order that we're publishing our papers. So I'm sorry that's really confusing. Bowland 2, it's actually called PNR 1. Bolan 1 is actually called PNR2, but this is what we're calling them in our papers. So the first well, we got 7 flowback samples that were collected from storage tanks. So it's fluid that had been collected early on, stored in storage tanks on site, because that's part of the regulation of how they deal with their fluids, and then tapped off for us to have a look at. They were also sat in our cold room for quite a while before we did anything with them. In contrast, when the second well was fractured, once flowback started occurring in October 2019, we were able to get samples straight from the well pad, straight from the gas water separator, and then we froze them all back except for the small amount of each that we kept aside for culturing. And then a month later we had a ban. So it's a very short window of opportunity. But the key thing to remember with these things, or to point out rather, is that the input fluid is very lean. So this is a comparison between the Marcellus that you've already heard quite a lot about and the Bolan. And these are a list of non-exhaustive list of additives that typically get added. So you've got a long list here for the Marcellus and a very short list for the Bolan. So in this case, it was a slick water fracture with polyacrylamide, not gel forming agent with guar. And they had permission to use hydrochloric acid, but I don't believe they did. And they used sand as a propellant, and that's it. So just to flag some of the key, patterns that we found, we see the salinity here. It increases to quite a high level in our first well, and it's already quite high in our second well, by about 100 days. It's lower, though. I realise I haven't given you a comparative number here, but it is lower in salinity to the US systems. These community profiles are done at a pretty high level because we wanted to fit all the information in, but it's really to highlight some of the key things. And the differences are that we do see major differences between the two wells. And there are some taxa between these two wells that are shared in common. And these have been pulled out here at family level. So the key ones to point out are Marinobacteriaceae, which we know actually in this case is Marinobacter. And you can see the blue colour, particularly in this right-hand one. It can be quite prominent in some of these bars. And Shewanella, listed here as Shewanellaceae. So this is the pink salmon colour. And this is definitely the most abundant organism in our system. So this is the work of Natalie Hernandez-Becerra. She's doing a PhD with myself and a colleague at the moment, and she's done a meta-study of 16S gene sequencing data and also the salinity data across lots of different wells to include ours in this more global view. So this now, for the first time, includes the Chinese system and also a UK system. So you can see the salinity is fairly similar in terms of its increase over time. You've got number of days along the bottom here. And you can see here that we've got some samples that were collected years after fracturing began. So they're real outliers in the usual subset. And then these second and third graphs just show that the circle or the oval, which is our samples from the Bolan, that we get a decrease in diversity as chloride decreases. And then we seem to have, compared to other systems, in terms of absolute diversity, so ASVs here you can think of as OTUs or as individual organisms or taxa, and we have a much broader spread than any other. It's fairly similar in terms of the number of ASVs here or taxa to the one in Canada. But if you see the Marcellus here, this is the one I was working on, by far the lowest diversity of these included here. And this is just a different way of looking at it. So in this case, the Shannon index of diversity, the higher it is, the more diverse. So they're similar, but ours sits here up with the highest mean there. And then just to put it into context, a more this is at family level here. This is a heat map to show you all of the different sample sites based on formation here. Along the purple gives an indication of how late stage these samples were collected. So you've seen already that we had a very short window of opportunity within 100 days, and that's where ours was really from. And every white line indicates a different well in that formation. So these are our two here. But one thing that I really want to highlight is this lack of it. So this is ranked based on most to least abundant across all of these sites. So the highest here, Halanarobiaceae, or Halanarobium as we know it to be, is listed at the top because it's so dominant in some of these systems and it's present in almost all of them. However, we find a complete lack of it, and there's a complete lack of it too in this Niobrara system. This is really interesting because we didn't expect that. But just to hone in a bit more on what we found in our own system, and to flag that we've got genomes that we're pulling out of the metagenomes from this right-hand well. We've got a number of different genomes of Shewanella, and we've been able to identify them to the species level. They're very complete, low contamination, and we find them at multiple time points. So the same genome we find over a time series, and we even find evidence of it in the drilling mud. So that's really interesting. Marinobacter we found to be very abundant, particularly in these late stages ones, these red ones here. And we found a couple of genomes, including a draft quality genome, which by the way is what this asterisk means. And again, we found that in almost all of our time points. So too did we for Rhodobacteriaceae and Flavobacterium. So we've got a really nice time series or time resolved metagenome analysis on the way at the moment. This is a non-exhaustive list, but it gives us an insight of what our most abundant players in this site might be doing. And this is the kind of work that I'm doing in the background here. Phylogenetic placement, who exactly are they, whether they're novel, what their metabolic potential is, in light of the fact that we know we only have polyacrylamide in this system. How are they adapting to the admittedly not as high salinity, but still high salinity? And do we see evidence of viral immunity and viral predation? But a key aspect of this work is to understand biofouling potential. This here is just a flag that we're not just getting these genomes recovered from the metagenomes, but we are actually able to pull them out in isolation. So this is what we think to be Marinobacter isolates that we're sequencing now with Sanger sequencing just to identify them. So we've got some really cool experiments lined up in the lab where we know who they are. So I'm just going to focus a bit more on one of these wells. In fact, this is the second well. We got very low DNA yields from this. So I take this information with a pinch of salt. This information that you're seeing in this graph is 16S gene sequencing data. It turns out the key differences between these two wells, which translate into this slightly noisy picture, is that there was no biocide added in these wells, but there was a UV sterilization system that they used, which is, by the way, a very expensive way of treating your potential microbial biofouling problems. However, we didn't get detectable DNA from this well at all. This is the dregs that we amplified up for PCR sequencing, which is a clear indication compared to other samples that the UV system worked really well. Nonetheless, we still see our Shewanella here, we see Gallionella, we see other things that are pretty typical, some that are not. So just to pull some of these out a bit more. This top subset graph is just to show the relative abundance of key taxa that we've identified in other previous studies. And we see Shewanella is by far the most dominant. And then along the bottom, we've got organisms or taxa that are phylogenetically indicative of sulfide production. Now, sulfide production is what we were mainly concerned about with this work because it's a long-known problem in the conventional recovery of oil and gas. So we were trying to understand what problems there were with this. But again, we're not just looking at the molecular information, we're also doing experiments. So this work was done by a PhD student who's submitted today, Lisa Cliff. She's also submitted her paper, which is what that means. And she did some nice microcosm experiments where she fed an enrichment culture from Boland 1 with guar gum and thiosulfate, but also polyacrylamide and thiosulfate, to see whether sulfide was produced. And we didn't find any sulfide production or thiosulfate reducing activity at all from polyacrylamide, but we did with guar gum. In fact, we found evidence for guar gum being broken down into these smaller organic compounds, which we think are then going on to fuel thiosulfate reduction. Not sulfate reduction. We don't really see sulfate reduction in shale gas systems. But then on top of that, we did find an observable level of sulfide being produced where we didn't find any in the control, which is clear evidence that guar gum can stimulate sulfide production. And in fact, this is something I've done some work on a few years ago too. And then because those experiments were done on the bench at ambient pressures, we wanted to understand what that might look like if we simulated downwell conditions. So with a really nice collaboration with Raw Water Engineering, our colleagues not too far from Manchester, they have these bioreactors that allow you to recreate the conditions of an oil and gas reservoir. So we did that with our system to try and get a sense of how the community would react to this same combination of thiosulfate with guar gum when we pack these bioreactors with sand and shale. So on the left, you see the control, which didn't have any inoculum added. although we do see some indication of biological activity. And then the two on the right, you can see the inoculated system, where we have Berlin shale communities inoculated. And the key thing here is we do see some attenuation of thiosulfate concentration. These are replicates, individual replicates. And we also see an increase in sulfate, which is really interesting. And we think this is thiosulfate disproportionation. Concomitantly, although this is noisy data, we do see production of sulfide where we don't see any in the control. So this is demonstration that only after 8 weeks, under simulated conditions for the downwell conditions, we do see the same effect. And we're going to be running these for a lot longer. So this is initial data that Lisa recovered for her thesis, but we're working on it in the background. And then crucially, the key aspect to the end of that would be to decommission these bioreactors, pull out the core from within, try and visualize some of those communities that might be associated with the shale under the microscope, and also do some DNA extractions. So we're monitoring the communities in these week by week with 16S sequencing, or we will be, and then we can also do some metagenomic sequencing on those. But one key unanswered question is whether the signatures in these flowback fluids are representative of what happens in situ. And you can't do that. You can't understand that question or answer that question in situ. You really need to recreate it in the lab. So we have a really unique opportunity here to address whether the community that's sessile is different, and whether there's particularly whether there's any potential for biofouling through biofilm formation. So I'm going to summarize that before I move on to my new research area. So hopefully you've been convinced that hydraulic fracturing creates new microbial ecosystems in the deep subsurface, and these really offer a window into life in these extreme environments. We know that fractured shales harbour novel organisms capable of withstanding all the pressures in these systems, so high salinity, viral predation, and other extreme conditions. In fact, the organism that I presented, the novel organism, isn't the only novel organism. You might have heard of Fracibacter, which is also an organism that was characterised as a novel genome from a novel genus from this environment. But the application of metagenomics is really conducive to uncovering unknown diversity. But understand that as studying the UK shale gas well fluids has highlighted a much broader diversity of microorganisms than we were aware of before that could potentially cause problems for shale gas extraction. Albeit we won't probably be doing that in the UK, but nonetheless these are becoming more and more important sources of energy for all of us. We buy much of our gas from the US now through their shale gas extraction. So understanding the potential of microbial problems in these systems can enable this process, inevitable as it likely is, to be conducted as efficiently and cleanly as possible. And that's really the goal here. So going back to our cross-section of the subsurface, and I want to move on to CO2 storage, because that's really where the work I'm doing in my group is pushing forward in. So here, we don't really understand what role carbon dioxide storage is going to have on the deep biosphere, but it's crucial to be able to permanently store that CO2 in the deep subsurface for carbon capture and storage to help us achieve our net zero goals. There are some critical knowledge gaps though. So we think CO2 injection will affect microbial diversity and function, but we don't understand the impact of that. Could be positive, so we might get a net drawdown of CO2 and a conversion to biomass, which could ultimately facilitate mineral precipitation, but they could also be negative, so there could be a net production of CO2 or indeed other gases that could affect storage capacity. Perhaps there'll be bioclogging and also corrosive potential. We also now understand that microorganisms have evolved to operate in communities, not in isolation. So the way that we do microbiology is changing. We need to understand community function and interactions between microorganisms. And by microorganisms, I don't just mean bacteria and archaea, I mean fungi and I mean viruses as well. And all of this presents a really unique opportunity for understanding whether we can harness processes in these communities for our benefit. And in this case, we as a group are looking to promote microbially driven CO2 sequestration in situ, but also to learn from CO2 driven communities about ways we can turn CO2 that we don't want into things that we do want, ex situ, biotechnological application there. And that really brings me to my plans over the next 5 years. So I'm going to continue to work closely with raw water in order to do this subsurface simulation approach. This will be used to simulate depleted hydrocarbon reservoirs and also saline aquifers. And these are all target formations to store our CO2 that we don't want in the atmosphere. This is complemented by high pressure cultivation, which we can do here in our labs, using communities that we've already been enriching from, for example, the Boland Shale, or from oil and gas formations that raw water have access to, but also new subsurface samples as well. And by understanding the environmental context through geochemical monitoring, we can really start to simulate CO2 injection and understand what that microbial response will look like under in situ conditions. And then the meat of this work is really to unpick that process, or that set of processes, to understand who's doing what and how it's doing it. So here we really want to be able to trace the fate of the CO2 through a community that can use it. And I will be broadening away from just using metagenomics, although genome resolved metagenomics underpins all of this work, onto which we can map transcripts, which tell us about what genes are being expressed. We can also monitor the proteins being produced and the metabolites being formed. And by tracing the CO2 through the community using isotopically labeled carbon dioxide, we can really pull out within that who's actively doing these processes and what the ultimate fate of that CO2 is. And in the end of all of this, if we can really carefully track, which we hope to do, the geochemistry of these systems and understand the flux balance, we can start to build conceptual models of community activity and turn those into numerical models. And this can be, the particular goal here is to try and understand or hypothesize new ways to produce useful things from CO2, but it also has lots of potential to be included in biogeochemical models where microbial community function is poorly represented or missing, and should also be useful in other systems. Perhaps as complexity builds, they might be useful in human microbiome, for example. This is me attempting to kick the whole project off. I'm in this picture 800 meters below the North Sea. This is in Bolby Potash Mine. This was a month or so ago. And I'm collecting fluid that is seeped down from the Bunter sandstone. This is a saline aquifer that actually NetZero Teesside project, which is a regional decarbonisation cluster involving major oil and gas or energy sector companies, I should call them. They hope to store the CO2 that they generate from splitting methane into water and CO2. They hope to store it there. So this is a chance to recover this fluid and understand the microbial community in there and how they respond. So we're hoping to do some culturing in the next few weeks to kick that off. You can see from this picture though, it's not very clean. This is fluid that has seeped into, this is actually a road that I'm standing on, a mine road that happens to be caving in a little bit, but I'm sure I'm not supposed to be showing these pictures. And this is polystyrene floating in it. So they're not clean samples, but so far it's the best access we can get. Although we are looking for other opportunities to access the subsurface. And I want to flag just briefly an opportunity to join my group as a PhD student. So we have a fully funded project that actually closes on the 31st of March for application, which is 2 days away. But this is the middle part of that big schematic I showed you, where we're trying to coordinate all these omics approaches, stable isotope probing, and high pressure cultivation, things like that, to really understand CO2 driven carbon cycling. I'm also looking for a short-term bioinformatics postdoc that could lead to other things. So if you know anyone, please direct them to me, or please reach out if you're interested in either of these. And I'm going to leave that up just for a second while I thank you all for your attention. Also just want to flag some collaborators of mine. So obviously the two students whose work I've presented today, Lisa Cliff, who's about to embark on new things, actually here in the MIB, and also Natalie Hernandez-Becerra, who's been doing some really nice work in the lab and on the computers. My close colleague, John Lloyd, who's been involved in a lot of this work, and then also the group in the US, Kelly Wrighton's group and Reb Daly. They've been really helpful in some of the early stuff and training me up. So I'll stop sharing my screen now and hopefully take some questions from you all. Thank you for your attention.